Okay, so today is the second lecture about ChIP-seq, and after that, next time, we'll be talking about epigenetic regulation, which is also ChIP-seq type of analysis. A little bit overview of what we have talked about last time, and the basic concept of ChIP-seq is ChIP-seq stands for chromatin immunoprecipitation following next generation sequencing. And so the goal here is to identify genome-wide binding patterns of a protein of interest. That protein can be a transcription factor, can be a histomark, or can be RNA polymerase. So you can see that this is double-strand DNA with protein binds on that. And the first step is to do the cross-linking. And these steps are important because we are going to use this information this time when we design or talk about other people design algorithm to identify the binding sites. So you do the cross-linking, which means the double-strand DNA will be binds on the protein, and then you kind of break those into smaller pieces, and then you use antibody to target this protein. And after that, you only sequence these sequence rates, and then using bioinformatics to identify where in the genome-wide that protein originally binds on. And we talk about the comparison between the ChIP-seq and the ChIP-ChIP. So ChIP-seq is actually not the first technology that we can do this genome-wide, and ChIP-seq was. But ChIP-seq is a higher resolution with the ones in bioinformatics analysis, and it has less noise compared to the ChIP-ChIP, and it's greater coverage. So this is quite interesting because we know about 48% of human genome is non-repetitive, which means 52% are. So those 52% are not going to be designed on the tiling array, so you won't get any signals around those regions. But if we use the ChIP-seq type of technology, and 80% of regions is mappable with the 30 base pair arrays, 89% with the 70 base pair arrays. The reason for that is some of the repetitive regions, they are connecting, they are borders of non-repetitive regions. So if a read cross that border, and then non-repetitive regions will help you to identify where they are. So even though they won't be put on the tiling array technology, but they are still being sequenced, and you can still map them back. Okay? And the ChIP-seq is currently much cheaper than ChIP-chip. This is another benefit. Actually, really much, much cheaper. And the shorter rates is an issue for assembly, but not as much for the ChIP-seq technology. Okay? Actually, ChIP-seq is the first real application in terms of in the research market about next generation sequencing technology. And we have talked about several studies that using this technology that enables something that we were not able to see before. And the first paper was published in Science in 2007, and that is the transcription factor binding mapping. That is one transcription factor, and which is very well defined. Their binding sites are very well defined. So basically, they want to use that paper to understand whether this technology will work or not. And the same year in Cell, there's another paper published by using ChIP-seq to understand the histone mapping. So we know that we will talk about those in detail next lecture. And there will be many different histone modification markers. And if you design the protein antibody to target those different markers, you can understand how the chromatin modification is really organized in genome-wide. And enhancer identification, and this is actually two papers published both in Nature, and they use the P300 as a marker, so they designed antibody to target a P300 that can help you to identify active enhancer markers. And we just briefly introduced that we can use this technology to study DNA methylation. And this is a review paper that you may want to take a look at. And they gave several different methodologies and biological methodologies to study DNA methylation. So we will also talk about that in detail next time. And there's two more studies to study actually not the protein DNA interaction, rather it's protein RNA interaction. And the technology is called the ChIP-seq, stands for cross-linking immunoprecipitation. And the first study was published by us. We are involved here, which is to study the protein RNA interactions, that is SR protein. And the second one is to target one of the proteins in the risk protein complex, which help us to understand the microRNA and mRNA targeting. So all these studies are kind of hallmark studies, and there's one thing I didn't mention here, which is also very important, which is the ENCODE project, right? And ENCODE project, which is the annotation of the human genome or several other model organisms, and ChIP-seq has been very extensively used in those projects. Today, we will get into the data analysis part. So we'll first give a very brief introduction of experimental considerations for the ChIP-seq experiment. So before you do the experiment, what do you think about that? What's the considerations you want to have? And for the analysis part, I'm going to talk about the peak identification, and this is really only for the transcription factors. And for the histone markers, that will be another different type of shape, and the methodology will be very different. We'll talk about it next time when we talk about epigenetics. And I'm going to talk very briefly about DNA fragment length evaluation, saturation analysis, and algorithm comparisons. And I'm going to give two brief introduction of integrated data analysis. The first one is motif finding related and related theories. And this motif finding is not directly related to ChIP-seq, but that is the natural step after ChIP-seq. So if you study one transcription factor, there are genome-wide binding patterns, you identify those regions, and after that, what are you going to do with it? You want to learn those regions in greater detail, which motif finding is a very natural step in that area. That's why I want to spend a little more time on that. And the last study I want to introduce today is identifying microRNA promoters using ChIP-seq data. This is one of the studies we published. All right, considerations in ChIP-seq experiment. The first consideration is the quality of antibody. When you do any experiment, you really want to get the high quality data. But for ChIP-seq, the most important factor is the quality of immunoprecipitation antibodies. And we have heard so many bad stories about ChIP-seq that after they spend so much effort to get the platform and get everything really figured out, and they found out that they didn't really immunoprecipitate down anything. And actually, we have some data that's sitting in our lab is in that situation, and which is completely wasted. Not only the sequencing cost, but personnel cost as well. And this is so I haven't personally worked on the data, the experiment by myself. But the issue is some of the people just assume that the Western, if some antibody works on the Western, it will automatically work on the ChIP-seq, which really doesn't happen in that way. This is a much higher quality antibody required. And if you want to test the assay before you go to the sequencing, you probably need a few positive regions. You run the qPCR after ChIP, after immunoprecipitation, run qPCR to see there are really positive signals, and then you put your samples on the sequencer to go to larger scale. But the point here is antibody is really, really important. And the second issue is you might also expect is the amount of initial material usually is very, very low. Okay? So if you think of DNA sequencing, okay, although you cannot get as many as you want, but you can get a pretty huge amount. If you're talking about RNA sequencing, that is still a lot of information, a lot of samples you can collect. But when you do the ChIP sequencing, you got the cell, and then you got the antibody to target that small region in the DNA. And that is such a small fraction of the entire DNA. So even though sometimes you start from a huge amount of material, you eventually will end up with very, very low input material for the sequencing part. And I think if you recall that two papers published in Nature about enhancer identifications, and if you think of the experiment, it's not that difficult, right? They did ChIP-seq, and then after that, they identify information out of it. But that is really difficult experiment to do. You got the mouse embryo at embryonic day 11.5. That is very, very small. And they have to pick one tiny region, forebrain or limb, of one tiny region of that small embryo, and then do ChIP-seq it's such a difficult experiment to do. I think that is additional credit given to them so that they can publish that in Nature. But the point here is, regardless of what you work on, I know some of the students working on the hematopoietic stem cells, and it's really, really hard to get a lot of initial material out of those. And I think one of the paper published that using ChIP-seq to study something in HSC, human hematopoietic stem cell, and they used 10 power 7 cells, all right, 10 million cells, HSC. I guess they just kill one person and get it done. And that's a lot of blood to go through to really get 10 million HSC from the human. And so those are something that sometimes can be a limiting factor for the ChIP-seq experiment. And because of this, when you do the experiment, of course, it's written in the standard ChIP-seq protocols that adapter usage is you need to really specially consider that. Use in ligation reaction should be reduced. So you got a limited amount of DNA, and you put adapters on there. If you use the same amount of adapter as for the RNA-seq or DNA-seq, you end up with a lot of things. It's just 2 adapters, right? And when you do the sequencing, you will see a lot of adapter or primer dimers, which doesn't have the real information inside. So I heard that initially when people work on the ChIP-seq experiment, they really have to spend a lot of time to polish their protocols, and eventually they were able to really put the fragments they want to put inside into the sequencing product. So those are something that you really need to consider. The third consideration will be the barcoding. So we know that sequencers, they are becoming more and more powerful. For each lane, Illumina lane, and I know they have high-seq technology, and they can produce many arrays. Initially, it used to be only 20 million arrays. Now for one lane, they can produce probably 200 million arrays. You don't need that many for one sample if you do ChIP-seq. But you still want to use that technology. How are you going to do that? You need to barcode your samples, which means that before you do the sequencing, you give special barcode oligos and put that in front of your sample. So you can see, you can get this for sample 1, and you put all AT there, and for sample B, you put all CT there. And after that, you mix everything up together, so put them together. And probably you'll put 2 samples there, and you sequence 100 million arrays, and each sample is roughly 50 million arrays. And then how you decipher that, you just look at the first 2 letters of your sample, and you'll be able to have a good idea which fragment is from which sample. So this is called barcoding or multiplexing protocol. And I think for the solid platform, you can barcode 16 samples, am I right? 16 samples. 96 is what they say. They said they are going to give us, but not yet, right?
No, it's 96 now.
It's 96 now? Okay. All right, so it's 96 now, but still I don't believe it. It's not going to be very high quality, but 16 is quite stable. Illumina is, I think, is 12 barcodes, but still they are making them more and more. Anyway, next consideration will be the pair-end technology. So we talk about this a lot. You've got DNA fragments, and you sequence both 2 ends of it. And the additional benefit of this is it helps you analyze the regions in the repetitive regions. This sometimes, if you work on the transcription factor and well-defined, it's probably okay. But if you work on the DNA methylation or histone modification, especially those histone markers that work in the hetochromatins, and there's a lot of repetitive elements that will be fished out. So you do the pair end, that potentially will help you a lot when you do the analysis. Okay? So this is the first batch of considerations you need to think through before you do the experiment. The quality of antibody, the initial material, barcoding, and pair end. And the next thing you want to consider is the so-called sequencing depth evaluation, meaning that We know these machines are really, really powerful. You can sequence 1 million reads for one sample, 10 million or 100 million, as many as you want. But how many reads do you really need for one sample? But this is not an easy question to answer, actually. It's condition specific, it's protein specific. It's really a kind of a question that there's no simple answer to give. And so people designed this so-called saturation analysis, and I'm going to give you a heads up here, and there will be a couple more slides about that in the later part. So the criterion here is after you do the analysis, you did the experiment, the results of the given analysis do not change when more reads are obtained. So this is the criterion. If this has been achieved, so let's say you spend 1 million reads, you identified 200 peaks, okay? And you spend 2 million reads, you identified 102 peaks. So that additional 1 million reads didn't give you too much additional information, and that suggested it's already been saturated, so you don't need to do more. Do more doesn't really help anymore. And but there's more considerations on that, and we'll get into there when we really talk about that. So the presence of a saturation point after which no further binding sites are discovered with additional reads, so you don't want to go further than that. And the next consideration is also the depth of sequencing. And so if we do the saturation analysis, which we'll talk about that, and it's already after the experiment. So after the experiment, you can evaluate how many additional reads do I need for this particular sample. But if before the experiment, how can I have a really fair estimation how many reads do I need for one experiment? And this is an equation published in 2010 that help you to make that judgment. Okay, and how accurate that is. This is the same author, the same group from the 2007 Cell paper regarding the histone mapping on the ChIP-seq, 20 histone markers, that study. And so they have a lot of experience. That's from Koji Zhao's group from NIH. So let's look at this equation in a little bit detail. I'm not going to tell you how they derived this. This is a little bit more complicated than that. So the first thing is that the S, which is the total number of mappable reads that are required or are desired, is determined by several factors. The first one is G. What is G? G is a non-repetitive genome size. Okay? For human, that will be 1.5 billion. Right? We know we have 3 billion letters in our human genome. Half of them are repetitive regions. And for this one, that will be 1.5 billion. And so the larger the genome size is, the more reads you will require to identify that. That's nothing surprising. So it's very reasonable. The second one is this W, which is the window size. Okay? So what that is meaning the resolution of the data you want to look at. If you want to get to the one base pair resolution, I want to know that for very, very fine detail that whether there's a binding or not, you do need a lot of resource to get through that. All right? If you say, I'm okay, I don't need the really one base pair resolution, as long as I have 100 base pair resolution, I will be happy. And then this W will save you a lot of money. So your total number of sequencing reads can be reduced by that. And the third one is the so-called E, which is the enrichment, the ratio of average tag number in the window containing a binding site to that in an unenriched window. So you can see that this E, let's say it's equal to 2. That means that you want this region, the enrichment is double the region that in the background region. So if there's one region it's not binding, this is a binding region, you want this binding region has 2 times more enrichment than the background regions. So if you think of it's 2, that is a pretty greedy type of looking at that, right? So if this part, there's no binding, you get 100 reads. With binding, you get 200 rates, and that is doubling it. And sometimes you need much, much more than that enrichment ratio. We remember that for the first science paper on that transcription factor binding, they require 5 times E is equal to 5. That's what they required. So which means that in the real binding region, they require 5 times more enrichment than in the background region. So of course, you can justify that in your calculation, right? So this equation, and once I put it here, it becomes pretty easy to look at, but they spend a lot of effort to derive it, and with a couple of assumptions. The first assumption is non-specifically precipitated tags are distributed uniformly and can be described by Poisson distribution. That's one of the assumptions. So meaning that there's no hotspot in the genome. So if there's a non-specific binding reason, they should be distributed equally along the genome, which doesn't really happen. And the second assumption here is the calculation is based on p-value 10 minus 4. Right? But nevertheless, this will be giving you a pretty good indication that before you do the experiment, how much resource do you need to sequence one sample. Next consideration will be the nucleosome resolution. So we remember that when we do the ChIP-seq experiment, your first step you'll do is the cross-linking, meaning if its protein binds there, make it binds there. And the second step is to do the sharing, to break the DNA into smaller pieces. And this is in that step, which is we call it fragmentation step. So there's 2 possible ways that you can break the DNA into smaller pieces. The first one is the so-called sonication. So basically, you use ultrasound to just give the additional pulse to the sample, and it will be broken into smaller pieces, right? What's the size of the fragments? That depends on how often you do it and how strongly you do it. So the biologists in the room probably know that machine pretty well, right? Got a tube, and something like that. I saw that in Dr. Edinburgh's lab all the time. But that's one way to do that, physically break it into smaller pieces. The second way to do that is based on the enzyme digestion, and which is a microcanonical nucleus and the digestion without cross-linking. So this is another way to do it. And but you want to have special considerations which way you pick, right? And the good thing of this use MNs to really to do the digestion is it removes the linker DNA more efficiently than sonication. So basically, if you treat you set your condition well, and this part will be break down into smaller pieces, and so your cutting will occur in those linker DNA regions. Okay, so that is the good thing of that, and why that is important comparing to the physical break it into smaller pieces. The reason for that is if you do in that way, you break only the linker regions, not the other regions, and you got your resolution down to 1 nucleosome resolution. Okay, 1 nucleosome meaning that this histone and the DNA wrap around it, and 1 nucleosome is 147 bases. Okay, and you got your final resolution down into 1 nucleosome resolution. And initially, people didn't think that is a big deal. It really doesn't matter, and I can do 200, 300 base pair resolution. Well, yes, that person is right. However, if with this type of treatment, if you're down to the 1 nucleotide resolution, and that will allow you to study something that higher resolution lower resolution cannot offer you. For example, this study in Nature Genetics, they found that this estrogen receptor, androgen receptor, and will make the histone markers move slightly. That when you treat with androgen receptor, so those kind of slight dynamics that you cannot really observe with sonication, break it into smaller pieces, and only this type of digestion will be helpful. And but the problems of this is it anytime you use enzymes to cut the DNA into smaller pieces, and then you are having the risk of have additional sequence bias, because we know that enzymes are not always cutting randomly from any place, right? It has their sequence specificity. Sometimes it will create additional noise. Again, for later, initially I didn't pay attention to about this, but now I read the 2007 Cell paper better, and at that time they already start to use this enzyme digestion. So their histone markers mapping really the resolution down to single nucleosome resolution, and I think it becomes a norm for the current reactions. And the next consideration I want to mention a bit is the control sample selection. Okay, and there's always a big debate whether you want to need to use control for the ChIP-seq experiment or not. Right? Of course, there are some groups they favor we need to use the control. We obviously need to do the control experiment, which most people will agree if you don't have to pay for the bill. And but we know that to do this type of control is really not very cheap, because you're sampling the entire human genome without any additional immunoprecipitation, and that has become really, really expensive assay to do. So let's go through the several types of controls that people are doing. And the first one is the so-called input control. Okay, so they will skip the step of immunoprecipitation. They don't use antibody to target on anything. Okay, and then they just sequence it. So this is the first thing, the input control. And the second one is called mock IP control, and which is you do the immunoprecipitation but without using any antibody. So you use a faked antibody. And this is another way to do it. The third one is non-specific immunoprecipitation, which is you do the immunoprecipitation performed using an antibody against a protein that is not known to be involved in DNA binding or chromatin modification, such as IgG. So you use antibody to target on IgG. You pretend it will bind on DNA, but we know it doesn't bind on DNA. And then you see how many non-specific binding events will be put down, so that can serve as your control. So basically, there's 3 ways to do this control. And the people who really pay for the bill, and they are concerning about that. This is too expensive to do it, right? And so there's another group, they will favor that we don't really need a control. And because of the first one reason is a large amount of sequencing is needed to avoid sampling bias. Because when you do the no antibody or whatever this type of control, you are trying to really just sample this part of DNA, that part of DNA. If you really want to sample it well, you probably need to do a lot of sequencing, and that is way too expensive. And another argument they have is you may not need those control if you want to compare 2 conditions. If you say that I want to have one condition, MCF7 cells, breast cancer cells, and before and after treating estrogen, and what is the estrogen receptor binding sites look like. And at this stage, you probably don't really need this type of control. Just comparing the peak you identified from both 2 conditions and to see whether there's a difference. So that will work as well. Okay? Any questions here? Okay. So we talk about the experimental considerations, and all these things it's just before the experiment. And the first one, so we just went through this a little bit, and we talk about the quality of antibody and the initial material, barcoding, and pair end, and sequencing depth evaluation. There's a couple of things, and the nucleosome resolution. So what step do you do for the fragmentation? You do sonication or enzyme digestion. And the last one is how do you do the control? So these are the considerations for the experiment. All right, any question? Yes? How long do I sequence? You are not talking about the DNA fragment length, but the initial small length, right? For the ChIP-seq experiment, I will see that 35 base pair will be adequate. And you can go to 50 base pair if you want. but make it longer, it only wastes money. Because you make it longer, it doesn't increase your mappability anymore, right? And so you probably, in terms of mappability, it pretty much saturates at about 50 base pair. You further increase it, it doesn't increase too much. And so you probably don't need more than that. Okay? Other question? All right, let's move on for the next, which is the data analysis part. This is the central part of this lecture. And the first topic I want to talk about is the so-called peak detection. So when we do the ChIP-seq experiment, the overall goal is to identify the regions that are enriched with the ChIP-enriched fragments. So this is the figure that I just screen copied from that first ChIP-seq paper published in Science in 2007. So you can see in this particular region that you are seeing there's so many different small fragments were initially put down by these ChIP reactions. And you can see a huge enrichment around this region, and therefore they will conclude that there is a binding site in this region. So no question about that. This is very straightforward, very simple. There's two considerations. The first one is the enrichment, how much risk I'm accumulating here. And the second one is the control samples. Keep in mind, in this experiment, they also data control. So you can see in this region, in control samples, they don't see a whole lot of signals. All right. That gives them additional confirmation that this peak is kind of initially is more on the protein binding driven. Okay. And if everything is this simple, this easy, we're done today, right? Just get the data and identify those peaks, and that's the end of it. But things are a lot more difficult than this. And when people start to look at data in greater detail, they understand the data structure is actually different. And sometimes it's unexpected. It all makes sense, but it's unexpected. The first one is when you look at the ChIP-seq signals, there's several different type of signals you always see. And this figure shows you the first type of signals. Let's go through it. They call it a punctate enrichment. Okay? So what happens is, for the top figure here is, if everything is above 0, that means those are Watson rates, meaning those are the sequencing rates you identify from positive strand of the DNA. And this part is from the negative strand. It's called Crick rates, right? Negative strand of the DNA. So if you accumulate them together, it will look like something like this, the total rates. But you can see a clear separation between the positive and negative strand. They form two different peaks. Okay, we'll talk about that in the later slides. And so what this type of pattern is a classical pattern for specific transcription factors, like so sequence-specific transcription factors. That's NRSF. This is the one that used in that Science paper, and CTCF, which is here, and STAT1. So these are all based on earlier experiments. So once you study a transcription factor, and that transcription factor has a clear sequence specificity, and this will be the pattern you always see. All right, we'll go through that why it's like that. And one binding site is represented by two peaks centered around the binding loci. Okay, this is the binding, the profile of one binding site look like. Right. And the second class of ChIP-seq signal is a mixture of punctate and broader signals. So you can see that this is one gene here, and the classical example will be RNA polymerase. So you can see that this is one gene, this is transcription starting site. Around the transcription starting site, you'll see this is one peak here on the positive strand and another peak on this negative strand, and they are shifting a little bit. And after that, there will be a less but broader region of enriched signals. So strong signals around the transcription starting site and active or stalled promoters, similar like the transcription factor binding sites signals, and then diffusely but detectable in the transcript region. So you can see in these regions, there's a lot of signals, but it will be much less than initially in the transcription factor binding sites. Okay? So this makes sense? But let me give you another thing that you may also notice, you don't understand why is that. Towards the end of the gene, there's another small peak of RNA polymerase. And this is, we always see this as well. Can some of the biologists in the room give me an explanation of that? Why that here, towards the end of the gene, and actually it's beyond the end of the gene, there is something that we see a strong accumulation of the ChIP-6 signals on the RNA polymerase as well. No? Yeah? Mm-hmm. So right. I'm not sure that's too much to do with the microRNA, but both of you are right. That this is the so when the transcription happens, the RNA polymerase start from the transcription starting site and do this kind of scanning, right? And it generates this big transcript, right? But it doesn't stop. The part will continue moving, and towards the end of the gene and even beyond the end of the gene. And there's some special mechanism to let the part2 stop. I think this is the region that part2 eventually stopped. So you always see this type of patterns when you look at the part2 signal. A big peak around the transcription starting site, and another peak that towards the end of the gene, and somewhere in the middle there's not too strong but still very rich signals. The third type of ChIP-6 signal looks like this. So this is, they call it non-point fashion, which basically is on the histone markers. Okay, so this is not transcription factor, not RNA polymerase, but somehow this is kind of enriched across a broader range of regions. And histone markers and other chromatin domain signatures range from nucleosome size domains to very broad enriched signals. So you can see this part of enriched signals is across several genes, and it's probably tens of thousands of megabase. So those are the third type of signals, and we are not going to talk about this in this lecture, and because this is epigenetic regulation, we'll talk about it next. But today, we only focus on the first type, which is the peak detection for the transcription factors. Okay? And so this is also called the peak finders. Peak finders are only for the first scenario, identify the binding sites of protein with very specific sequence preference. The basic steps here is, there's several different steps. The first step is a signal profile definition along each chromosome. So basically, whatever algorithm you use, you first scan the genome about the ChIP-6 signals and trying to find some candidate regions that potentially are the binding sites. And the second step is we need to look at the background model. So meaning that if there's no binding sites, what the ChIP-6 derived signals should look like. Okay? And this can be control comparison of statistically derived model information. So we'll get into that. And after that, there's a post-call filtering. And basically, you want to remove the artifactual peaks. And this is really based on the people's experience during the past couple of years. The people know more and more about ChIP-6 data, what that looked like. And they figure out some of the strategies to do the post-call filtering. And the last step will be the significant ranking of called peaks. So if you say there's a peak, a ChIP-6 derived peak in this region, you better tell me how significant that is. And this is the last step. And another thing is good or bad is so far there's dozens of algorithms are available. So in this lecture, I'm not going to tell you any one of those specific algorithms, how they work, and it's not the purpose of this. Instead, I'm going to give you a summary of the general steps that people are following. So some of the part may be applied to one algorithm, some others may be picked from other algorithms. Okay, so the first step is to look for the enriched regions. And this is kind of very straightforward. So you see, I'm bringing up this picture again. So you see this is one region that you identified a lot of signals, and then you want to plot in this way. So each Y here corresponding to the number of reads you detected in that particular genomic loci. So you see there's a strong peak around this region, and that tells you, oh, there's a binding size. So this is a very straightforward way to do this, which is called counting. So this is take a direct sensors of mapped sites along the genome and allow very contiguous set of base pairs with more than a threshold number of tags covering them. So just basically you count the numbers and then you set up, oh, it's beyond a certain threshold. This is the peak. All right, very straightforward. And the good thing of this is easy and straightforward. The bad thing is it only works for the regions with very, very strong ChIP-seq enrichment signals. And for anything that's shorter than that, you wouldn't see very confident peak out of this. So most algorithms will take different strategies, but before we go to the different strategies, let's revisit this ChIP-seq protocol a little bit further. So we talk about there's several different steps. The first step is to do the cross-linking, which is, you see this protein and double-strand DNA, they're binding here, right? And the second step is to do the sharing. The sharing happens just randomly. It can be shared from here, and any single particular location. So you can see this sharing happens on those yellow arrows. And the third step is to do the immunoprecipitation. Next step is to do the sequencing. But the features of this is the fragment length of this DNA after you share it is larger than sequence read size. The sequence read is only the very beginning of it. So you can see this is a DNA fragment. Let's say this is 200 base pairs, and you only sequence the first 35 or 50 base pair. Okay? And the second feature here is the strand-specific structure of the tag distribution, which means if you see there's a red line, that is the fragment from the positive strand, and the blue is from negative strand. So we have a pretty much equal distribution of red and blue, right? But keep in mind, when we do the sequencing, we can only sequencing the reads from the 5' towards the 3' end, okay? So once we capture this fragment, we can only sequence this end. We cannot sequence this end. Okay. The same thing, if we capture this fragment, which is on negative strand, we can only sequence this end, not this end. That means that for a good signal, that all the reads that on the positive strand is on the left side of this protein binding, and the negative side will be on the right side of the protein binding. Does that make sense? Let's hope this figure will tell you a little bit more about that. So you will observe two separate peaks of tag density on positive and negative strands. So you can see here is if this is the binding size, the middle part is the binding size, you will see a strong peak of the red signals along this side and the blue along this side. Okay? This is just simply because your sequencing fragment is about a couple of hundred base pair long. You only sequence the very beginning of it, and your sequencing can only occur from 5' towards the 3' end. And the distance between these two peaks, actually reflect the size of the protected region, which is the protein binding site region, and also influenced by the size distribution of the DNA fragments. Okay? All right. So most of the peak identification algorithms are actually taking advantage of this particular principle. So the first strategy that people do is called tag shifting. Okay? So what they do is they will shift half of the average DNA fragment size. For the ones that detected on the positive strand, they will shift towards the downstream side about half of the DNA fragment length. So we mentioned in the earlier slides, the DNA fragment length can be 200 base pair or can be something else. Okay, and we will talk about how to decide that DNA fragment length in the future sections. But here, the tag shifting is once you got the positive strand signals, you shift it half of the size, and then for the negative signals, you shift it half size towards this left side. And after doing that, you will see a much stronger peak signals that just right, hopefully it's just right on top of the real binding sites. And the DNA fragment size can be identified computationally, and we will go through that in the later slides. So you can see that I'm going to use this board. So this is the forward strand raise, and this is the reverse strand raise, right? And once we do this shifting, your data will look like this. And if we do not do the shifting, we just add them up together, what you see is this yellow line here. So this will tell you that this tag shifting will increase the resolution slightly and increase your peak identification, the signal intensity slightly as well. So this is the first step, first strategy people do because of this particular protocol, and which is called tag shifting. Any question here? Yeah.
A lot of transcription factor dimerize that affect this because it's going to protect a larger space than what you're.
That's a very good question. That's a very good question. We'll have one slide to talk about the problem of this. And but that's a very good question. If you see that type of shape, and that will be we have to be cautious regarding the interpretation. Any other question? No? So this is only for single peak time, as you see. And the second strategy to do this is to do the peak, to do the extension. And this is some of the software we are familiar with doing, which is including the Partec and several other strategies. We start evaporating again. So this is called XSET, which is the extended ChIP-seq tag along their strand direction, and then count overlapping above a threshold as a peak region. So what happens is once you map those small signal rays back into the genome, and then you just extend that, say I'm going to extend to the original DNA fragment length. Let's say it's 200 base pair. Let's just extend it 200 base pair. And then when we count the numbers, and I use after extension rays length and to count the numbers. Okay? And the advantage of this tag extension before signal detection is it corrects for the assumed fragment length and it smooths over gaps that were not tagged because of low sampling and read mappability issue. Because if you only work on this area, so because we know that these fragments are pretty short, it's a 35, 50 base pair, it's very possible that some of the regions you don't see any signal at all because it just happens there's no tag there. Or, this is not my computer. What is that? I'm not chatting with somebody. No, that's fine. I'm going to end it. I'm kind of afraid that recording thing that doesn't work, so people are not here, doesn't get it, which is fine. So what happens is because of this short read feature, so you may see there's some part that doesn't have any fragment at all if you don't do the extension. And then you're thinking there's two interpretations for that. The first one is there's no signal. There's really no signal. But the second one is we just didn't sequence deep enough. If we did deep enough, we probably would get something. Or this particular small region happens to be not mappable, because it's overlapping, it's the same sequencing with somewhere else. And that is causing there's nothing mappable here. So once you do the extension, and those can be smoothed out, so you don't have to worry about those issues anymore. Okay? So because of this feature of this protocol, how this works, and keep in mind the two possible strategies. The first one is tag shifting. The second one is tag extension. Okay? They both have their own advantage and disadvantages. All right. Another thing that most algorithm is doing is called a sliding window strategy. So if you go back to these slides, go back to this. If you only do this kind of shifting or whatever, and then you count the number of arrays that covered each individual location, and you may end up see that there's a very kind of spiky type of features, right? And so this region you can see is up and down and up and down, up and down. And those doesn't really mean anything. But once you look at the signals, you just look at this way. So once you do the sliding window strategy, which means that within a window size, you can decide the window size. Replace the tag count at each site with the sum value with the window centered at the site. So just give every single nucleotide in the same window the same value. And that will denoise your signal a lot. We'll give you an example of that, and which means this is smoothing the signal. The second advantage of this is it can bring in broader statistical tools, which means that within each individual window, that by definition, the number of sequencing reads you detect can potentially follow the Poisson or negative binomial distribution. And then you have a lot more statistical tools can help you to identify the real signals. And there's many algorithms using this strategy, and how you will help you to find out the publications of all this, I'm hoping. And this is one of examples to show you the performance of the smoothing. So you can see this is a ChIP secret. I don't know exactly which protein it is. It's from one of my friends from Ohio State. So you can see this is before smoothing. You will see at each individual location there's a very spiky type of signal. But after you do this window size smoothing, and the signal become like this. All right. So if you were an electrical engineering background, which I was, and this type of strategy is very common, and people use that a lot. All right. I think we'll take about a 5 minutes break at this point, and we'll resume from there.